हेलो मैम। हेलो सर।
Mana ada problema?
Hello, sir. Hello.
హలో
మేడం
వినిపిస్తుందా?
వినబడట్లేదు మేడం, వాయిస్ మాట్లాడుతుంది.
ఇప్పుడు వినిపిస్తుందా?
ఓకే మేడం, ఇప్పుడు వచ్చింది వాయిస్. స్క్రీన్ షేర్ అయింది మేడం, ఇప్పటిదాకా వాయిస్ లేదు.
ఫస్ట్ నుంచి స్టార్ట్ చేయాలి. So today we are going to learn genome, as we couldn't hear the audio till now. So I'm brushing it up again. So the topic is genome, and the main objectives are that we learn all these topics from this unit. So let's get into the introduction. So eukaryotes. Eukaryotes are the organisms whose cells contain complex structures inside the membranes. So when you see the primary distinction between the eukaryotic cell and a prokaryotic cell, you see that is the eukaryotic cells have a membrane-bound nucleus, whereas prokaryotic cells do not have a membrane-bound nucleus. Most of these eukaryotic cells, they contain membrane-bound other cells like mitochondria, chloroplast, Golgi apparatus, and all the species of larger animals or larger organisms are considered as eukaryotes. Next, this is a eukaryotic cell and the constituents of the cell. Next, concepts of genome organization. Primarily, nucleus is called as a control center of the cell. Why it is called the control center of the cell is that its function is to maintain the integrity of the genes, and also it regulates the cellular activities by regulating the gene expression. By regulating the gene expression, it controls the cell activities. So it is called as control center of the cell. The organelles inside the cell they contain chromosomes. So what are chromosomes? Chromosomes are thread-like structures which are located inside the nucleus, and they are generally made up of protein and a deoxyribonucleic acid, which is DNA. Chromosomes are made up of proteins and a single cell molecule of DNA. Generally, the nucleus structure, it contains nuclear membrane, nucleoplasm, chromosomes, and nucleolus. It contains nuclear membrane, nucleoplasm, chromosomes, and nucleolus. This is the anatomy of the nucleus, structure of nucleus. Genetic material. So, as we discussed, the DNA is present in chromosomes. So, the cell's majority of genetic material is present in the form of chromosomes. When during the cell division, you find chromosomes. But during the resting period, when there is no division of the cells, when the cell is not in a dividing state, which is called as resting phase, in this, you see the chromosomes organized into long thread-like structures which are entangled. These are called as chromatin. It is present in the form of chromatin, and during the cell division, it will be in the form of chromosomes. There are two types of chromatin, euchromatin and heterochromatin. What is euchromatin? The less compact chromatin, and it also contains frequently expressed genes. In the euchromatin is less compact and it has frequently expressed genes. Whereas in heterochromatin, it is more compact when compared to euchromatin, and it generally has infrequently expressed and transcribed genes. Heterochromatin. It is of two types, constitutive heterochromatin and facultative heterochromatin. What is constitutive heterochromatin? It contains chromosomal structures like telomeres and centromeres. Telomeres and centromeres are present in constitutive heterochromatin. What is facultative heterochromatin? Generally, genes are organized into this heterochromatin only in certain developmental stages. Only in certain developmental stages, genes are organized as facultative heterochromatin. The name facultative indicates it. Chromosomal territories. So during the interphase of cell division, the chromatin organizes itself into individual patches. Chromatin organizes itself into individual patches that are called as chromosomal territories. Generally, the active genes present in this chromosome, they tend to move towards the territorial boundary. These are chromosomal territories. Next is chromosome structure and organization. Chromosomes, as we discussed, they are the complexes of both DNA and proteins. The cells contain more than one type of chromosome. Not only nucleus contain chromosomes, but also mitochondria and chloroplast also contain their own chromosomes. Mitochondria in both plant and animal cells and chloroplast in plant cells contain their own chromosomes. These DNA molecules in the chromosomes are circular in shape in prokaryotes and linear in shape in eukaryotes. So in prokaryotes, you find circular DNA, whereas in eukaryotes, you find the linear DNA. And they are composed of 10,000 to 1 billion nucleotides. Cells contain more than one type of chromosome. into chromosomes. In 1970s, techniques of biochemical assay and electron microscopy were used for this to know how DNA is packed into the chromosomes. In 1973 and 1974, a new assay was discovered, which is nucleus protection assay. In this experiment, what you do is you use a nuclease. Nuclease, we all know that nucleases, they cut the DNA. So these nuclease is used and it is the chromatin complex is treated with this nuclease and it cuts the DNA at specific locations. These are generally the locations where the DNA is not protected by any protein. So what does this assay indicate? This assay indicates the protein positions on the original DNA. From this analysis, you can see that proteins appear as beads on the chromatin. They appears in the form of beads, and each bead contains 8 histone molecules. So this protein appears as beads on the length of the chromatin, and each bead contains 8 histone molecules. What are these histone molecules? 2 each of H2A, H2B, H3, and H4. These 8 histone molecules. They are arranged in the form of a barrel shape. So they are arranged in the form of a barrel like this. So these 8 histone molecules are arranged in the form of a barrel, octamer shape, and it is bound by 2 DNA strands. It is bound by 2 DNA strands around it. This structure is generally called as beads on a string structure. Beads on a string. When you unpack the tangled chromatin, when you unpack it, then you can see the string. See the string-like structure on which you can see the beads. And they also discovered some 30 nanometer of fiber. So 30 nm of fiber was discovered. In this discovery, they said that, or they thought, the nucleosomes might be held together by a group of additional histones. In 30 nm fiber, it was thought that the individual nucleosomes might be held together by a group of additional histones. These additional histones are called as linker histones. These additional histones are called as linker histones. And the structural studies supported the traditional method. The structural studies supported the traditional model. So this is how the eukaryotic chromosome is organized. You can see the DNA double helix, and then DNA is wrapped around the histone, octamer histone, and then nucleosome is coiled into chromatin fiber. This is the further condensation of the chromatin, and this is the duplicated chromosome which you see in the metaphase. So what are the special features of this metaphase chromosomes? Generally, you find chromosomes during the metaphase. After the replication, after DNA replication, you see 2 copies of the chromatids, which are held together by a centromere. This is generally seen in the metaphase. And the arms of this chromosome are called as chromatids, and 2 of them are called as sister chromatids. The terminal structure of this chromatids are called as telomeres. These telomeres are of different lengths. They vary in the lengths. And individual chromosomes generally can be recognized By the length of the chromatids, the location of the centromere, you can recognize the individual chromosomes. Next is the chromosome banding technique. What is chromosome banding technique? So you stain the chromosomes here, and after staining, you find some banding patterns. So these banding patterns, they are characteristic to different chromosomes. So this is chromosome banding technique. You stain it and you find different patterns, and each pattern is characteristic to different chromosomes. This is a metaphase structure of a metaphase chromosome. These are the sister chromatids. So this is the karyogram. This is a pictorial representation of this banding technique. So we saw the chromosomal banding technique, right? So this is the pictorial representation of these chromosomes. How you find them? Each of them are different, and every chromosome has their characteristic length and centromere. Next. So there are also some unusual features you see in some other organisms except humans. Other than all the genetic material we have seen now, there are also some unusual features that are displayed by the organisms except humans. They are mini chromosomes. So the name indicates mini chromosomes are short in length and rich in genes. This example is chicken gene. And the next is B chromosome. What is a B chromosome? The fragment. The fragment. So during unusual events and during the cell division or nuclear division, there might be some unusual events because of which you see some fragments left over. These are called as B chromosomes.
Hello, madam. Okay, one second.
So these are called as B chromosomes. The fragmentary versions you see after the unusual events of nuclear division are called as B chromosomes. You find them generally in plants. Next is holocentric chromosomes. Holocentric. They are not only one centromere is present, but there are multiple centromeres found in this chromosomes. It is called as holocentric chromosome. Example, nematode. Nematode, in nematode worm, you see multiple centromeres, which is called as holocentric chromosome. So here you can see the fragments left over after nuclear division. These are P chromosomes. And this is, here you can see the difference between monocentric chromosome and holocentric chromosome. In monocentric chromosome, you find one centromere, whereas in holocentric chromosome, you find multiple centromeres. Next, we'll see about DNA protein interaction in centromeres and telomeres. So these two regions generally contain repetitive sequences. So these contain so many repetitive sequences. And in Arabidopsis and humans, generally they display basic centromeric pattern. So they both have almost similar and basic centromeric pattern. Whereas in yeast, Saccharomyces, here you observe it is defined only by a single sequence. In yeast, you find single sequence. So this is the difference between other eukaryotes like Arabidopsis and human, and only in yeast you find this single sequence pattern. Centromeric DNA region is much longer. This is generally much longer, and as we discussed, it has many repetitive DNA. In all the eukaryotes, except this yeast, because in this yeast you find only single sequence, so you don't find any repetitive DNA in this yeast. And here the binding site is very small because of this reason. So telomeres, we all know telomeres are the territorial parts. So these telomeres, they are the ends. So territorial parts are the ends of the chromosome. What does they do? They distinguish the real broken ends and unusual broken ends. So this helps in where you can say when there is unusual broken end, if the telomere recognizes that is an unusual broken end, then it can heal itself. If it is a real end, then it don't heal. For this distinction, telomeres help. These telomeres also contain repetitive DNA like centromeres, and they are special binding proteins. There are many repetitive DNAs and special binding proteins in telomeres as well as centromeres. Generally, the human telomeric DNA is made up of hundreds of copies and repeated motifs of this pattern. 5 motif, TTA, GGG, 3 end, with a short extension of 3 terminus. So from 5 terminus to 3 terminus, you'll have this repeated motif. And there are 2 special protein bindings. As we discussed, telomeres have repetitive DNA and also special binding proteins. So the repetitive DNA has this motif, and 2 special proteins here are TRF1 and TRF2. What is TRF1? TRF1 helps to regulate the length of the telomere. TRF1 helps to regulate the length of the telomere, whereas TRF2 maintains the single strand extension. It maintains the single strand extension. So this is the use of 2 special proteins that are present in the telomeres. And the proteins, there are also some other proteins present in telomeres other than these 2 special proteins. They are called as linkage proteins or whatever. What they do, they link between the telomeres and the periphery of the nucleus. They act as a clamp or they link between telomeric ends and the periphery of the nucleus. These proteins also helps in mediating the enzymatic activities. As a role of protein, it helps to mediate the enzymatic activity. So this is the telomere. This is the telomere. You can see here. And this is a repeated motif. You can see. Next. Genome and gene organization. So the genes within the nuclear genome. We'll learn about genes within the nuclear genome. So all along the length of chromosome, you don't find genes. Genes are not arranged evenly all along the length of the chromosome. They're randomly distributed where you see somewhere you find on the length of the chromosome the density is more, and somewhere you find the density is very less. Somewhere you don't find genes. So it's like that. It is randomly distributed on the chromosome. And this density varies from 1 to 38 genes. So this varies from 1 to 38 genes per 100 kb length of DNA in higher plants, whereas 0 to 64 genes per 100 kb in human chromosome. In human chromosome, 0 to 64 genes you find in 100 kb length, whereas in higher plants you find 1 to 38 genes per 100 kb length. So we use GSMA stain. So GSMA stain, it has a greater affinity to DNA regions that are rich in AT bases. So wherever you find AT bases, this GSMA stain has greater affinity to DNA at that particular base. So the dark G bands in the human karyogram are AT rich regions of the genome. So we have already seen the chromosomal bands. If you observe that, you find some darker regions in that. Representing the AT bases, because generally the AT contents in the chromosome might be like 45 to 50%. So when you see there, you can see the bands clearly, which occupy about 45 to 50% around. So in staining, in chromosomal staining, we use GSMA stain. Chromosomal staining for band pattern, we use GSMA stain. And gene organization, again, in nuclear genome, we are studying about it. So the differences in the gene organization, it varies between the eukaryotes, and it reflects the important distinction between genetic features and evolutionary histories. So it is important to learn about this, to know about the genetic features and evolutionary histories. Genes generally make up only very small part of human genome. Genes make up a very small part of human genome. For example, if we take 50 kb segment of chromosome 12, if we study about 50 kb segment of chromosome 12, what all does it have? It contains, first, it contains 4 genes. They are PKP2, SYB1, some unknown gene, and CD27. So what is PKP2 on this chromosome 12? This is which codes the plakophilin. This plakophilin is a protein which is involved in the synthesis of desmosomes. Desmosomes are the structures that act as connection points. These act as connection points between two cells. So PKP2 codes for plakophilin, which is a protein involved in synthesis of desmosomes. SYB1. SYB1 specifies a vesicle-associated membrane protein. It specifies a vesicle-associated membrane protein. And some unknown gene. It is named as FLJ10143. And next is CD27. CD27, it codes for a member of tumor necrosis factor receptor. It codes for a tumor necrosis factor receptor. And apart from 4 genes, it also has 88 genome-wide repeat sequences. It also has 88 genome-wide repeat sequences. There are 4 main types of genome-wide repeats. They are lines, long interspersed nuclear elements, signs, short interspersed nuclear elements, LTR, long terminal repeats, DNA transposons. These are the 4 main types of genome-wide repeats. And also it contains 7 microsatellites. Microsatellites, these are the sequences in which a short motif is repeated in tandem. There is a short motif and it will be repeated again and again. This 7 microsatellites. So one of these microsatellites has a motif of CA. The first microsatellite is CA and it is repeated 12 times. Same motif is repeated 12 times. And the other microsatellites are, other 6 are CAAA, CCTG, CTGGGG, CAAA, TG, TTTG. These are the 6 other microsatellites. So 30% of this 50 kb, the other 30% of this 50 kb segment which we are talking about is made up of stretches of non-genic, non-repetitive, single copy DNA which has no known significance. So as we discussed, the genes make up only small part of the human genome. This is an example. The 50 kb segment is an example where you see only 4 genes and the rest is gene-wide repeat sequences, microsatellites, and 30% of it is of no significance. These are the main components of human genome. Next, C-value paradox. C-value. C-value is the amount. This is generally denoted in picograms. C-value is generally denoted in the picograms. The picograms of DNA contained within a haploid nucleus is called as C-value. The picograms of DNA contained within a haploid nucleus. A haploid nucleus which contains certain amount of picograms of DNA is called as C-value. Organisms with higher DNA content are not necessarily complex. So if they have high DNA content, it doesn't really mean they are complex. For example, plants have more DNA than humans. So that doesn't mean plants are more complex than human, right? So organisms with highest DNA doesn't represent that it is complex. Two closely related organisms can have very different DNA contents. So if you see two organisms very closely related, but still they might be having different DNA contents. So this is stated as C-value paradox. What is C-value paradox? The amount of DNA in the haploid cells of an organism which is not related to evolutionary complexity. Simply, we can say two closely related organisms doesn't have similar DNA constitution. This is called C-value paradox. So it doesn't depend on the evolutionary complexity. The genome-wide repeats, these play a major role in saying about the compactness of a genome. It represents the compactness of a genome. So here you can see the eukaryotic genome sizes. They have different sizes. So the size doesn't mean the organisms are less or more complex. And this is sizes of eukaryotic gene. Where you see bacteria has 940 MB and Homo sapiens has 10 genes per MB. So it varies. Next, kinetics of DNA reassociation. So now we'll see what is DNA reassociation. So the sequence complexity has been analyzed in eukaryotes by reassociation kinetics, by restriction mapping, and sequencing of DNA. By using this reassociation kinetics, restriction mapping, and sequencing of DNA, the complexity of eukaryotic DNA has been analyzed. For example, Human Genome Project. These all made the studies of sequencing quite clear and simple. So we'll see about DNA reassociation technique. What we'll do in this, until there is a denaturation. So until it denaturates into a single stranded form, you heat it. And after that, you then slowly cool it down. So while cooling, the strands can pair back together. While heating, the genomic DNA denatures into single stranded forms, and then by slow cooling, you can see the strands can pair back together. While the sample is cooling, the measurements will be taken. Which measurements are taken? You see that how much of DNA is base paired at each temperature. You cool it very slowly so that you will see at particular temperature, you will see certain amount of DNA is paired, certain bases are paired. So you note it down. At what temperature, which DNA base is getting paired. The amount of single and double stranded DNA is measured. How do you measure the amount of single and double stranded DNA? You measure it by rapidly diluting the sample. So by rapidly diluting the sample, it slows down the reassociation of the DNA. and then binding the DNA to a hydroxylaptate column. So in hydroxylapatite column, you bind the DNA and you slow down the reassociation by diluting the sample. This column, you first wash it down with a low concentration of sodium phosphate buffer. The column is washed down with a low concentration of buffer. And with this washing, you can elute out the single-stranded DNA. And then with high concentration of this phosphate, you can elute the double-stranded DNA. At low concentration, this elutes the single-stranded DNA, and at high concentrations, it elutes the double-stranded DNA. So the amount of DNA in these two solutions is measured. So separately, you can measure it using the spectrophotometer. COT analysis. So this is a technique which is based on the principles of DNA reassociation kinetics. So this is a biochemical technique that is based on the principles of DNA reassociation kinetics, which helps us to measure how much repetitive DNA is in a DNA sample. So in a genome sample that you have taken, you can measure how much repetitive DNA is present. This is called as COT analysis or C0T analysis. So this is used to study the genome structure and organization. This COT analysis is used to study the genome structure and organization. So this is the technique for detecting the repetitive DNA. So here you shear it, you heat it, you denature it, and then reassociation by slow cooling. You can see some single and double strands. And then you pass it out to this column and you see single-stranded DNA. And here you see double-stranded DNA. And that's how you collect it and keep it under the spectrophotometer for the measurement. So analysis of this procedure. What you see from this procedure? From this procedure, you find how much repetitive DNA is present in this genome. And the rate at which the sequence will reassociate is proportional to the number of copies of that sequence. Here, sequence will reassociate at the rate at which a sequence will reassociate is proportional to the number of copies of that sequence. So generally a sample with high repetitive sequence will renature very fast. While when you have complex sequences, then it will renature very slowly. So the amount of renaturation is measured with a value called COT value. The amount of renaturation is measured by a value called COT value. In this, CO represents the initial concentration of DNA and T represents the time in seconds. CO is the initial concentration of DNA and T is the time in seconds. So the repetitive DNA will renature at low COT values. At low COT values, that means in less time, you see higher DNA. So this you have low COT values, while complex and unique DNA sequences will need renature at higher COT values. That means it will take more time to renature. So the more the COT value, the sample have higher repetitive sequences. The faster renaturation of this repetitive DNA is because there is available of more complementary sequences. You have more repetitive DNA so that it will take less time to renature. So fast renaturation can be seen. Application. So application of this to the genome sequencing. COT filtration is a technique which uses the principle of DNA renaturation kinetics. So this we have seen, and this is used to separate the repetitive DNA sequences. This allows DNA sequencing to concentrate on the parts of the genome that are more informative and interesting. So this COT filtration, it allows the DNA sequencing to concentrate on the parts of the genome that are more informative and interesting. So what will it help in? It helps in speeding up the discovery of new genes. So when the discovery of new genes and new experiments this will be quite helpful to by allowing the DNA sequencing in the interesting parts of the genome. So this is the COT analysis. So as we said, this is a % single strand DNA. This is the time. So repetitive sequences and single copy sequences. Here you see single copy sequences. Here you see repetitive sequences. So at lesser time, you find repetitive sequences. In more time, you find single copy sequences. So other important aspects of genome. So every eukaryote has many multiple copies of genes, and some among them are ribosomal RNAs. So other than the normal genes that we have discussed till now, we also have certain other groups of genes, and we'll discuss about this in this chapter. So some of them are ribosomal RNAs. These genes are present in multiple copies due to heavy demand. So there's heavy demand for the ribosomal RNAs. So they are present in multiple copies, and you find many ribosomal RNAs. These ribosomal genes are example of classic multi-gene families, and there is a thought that it might have been arisen by gene duplication during the evolutionary process. So there is some theory saying that, or some studies say that RNA genes might have arisen by gene duplication during the evolutionary process. So this is ribosomal RNA. Next is mammalian globin genes. So we all know globins are the blood proteins, and these combine to make up the hemoglobin in our blood. So in humans, we find two types of globins. The first type of globin is generally coded by a small multi-gene family on chromosome 16. The first A globin, it is coded by small multi-gene family on chromosome 16, whereas second A globin is coded by a second family on the chromosome 11. These are again the first one is ribosomal RNAs. Second type of genes are mammalian globin genes. Third one is pseudogenes and other evolutionary relics. The human globin genes have 5 inactive genes. In this globin gene cluster, you have 5 inactive genes. These inactive genes are called as pseudogenes, and these are also might be due to the evolutionary process and indication that genomes are continually going change. So it might be during the evolutionary process, they might have produced some pseudogenes. This is called evolutionary relic. So there are two types of pseudogenes. First one is conventional pseudogene, and second is processed pseudogene. What is conventional pseudogene? Inactivated gene because of its nucleotide sequence, and this might be due to the change by mutation. So inactivated gene because of nucleotide sequence, because of its nucleotide sequence. This change in the nucleotide sequence might be due to the mutation. During the change in the nucleotide sequence, which results in the inactivated gene. This is called as conventional pseudogene. What is processed pseudogene? This arises due to abnormal add-ons to the gene expression. So there might be some add-ons to the gene expression, abnormality, and you find the processed pseudogenes. And here you can see the formation of classical pseudogenes and formation of processed pseudogenes. By add-ons, you find processed pseudogenes, and by some mutations in the nucleotide sequences, there might be the production of inactivated genes causes conventional pseudogenes. Next is repetitive DNA. Next category is repetitive DNA. So you find many DNA sequences again and again and again repeating in the haploid genome. These are called as repetitive DNA, which are found more than once are called as repetitive DNA. And these might have some regulatory and structural functions. These repetitive DNAs might have some regulatory and structural functions. This repetitive DNA acts as repository from which new functional sequences can be obtained, and it also helps in production of abundant gene products. So they act as both repository from which new functional sequences can be obtained, and it also helps in the production of abundant gene products. So that's it. It helps us repository. And based on the number of times the DNA sequences are found, there are two types of sequences. The repetitive DNAs are of two types based on the number of times the DNA sequences are found. First is moderately repetitive DNA and highly repetitive DNA. Transposable elements. These are the elements. After repetitive DNA, the next category is transposable elements. These are the inserts which can insert themselves at the sites throughout the gene. These elements can insert themselves at any site throughout the gene. So there are two types of transposable elements. First is tandem repeats. Second is interspersed DNA. First is tandem repeats and second is interspersed DNA. Tandem repeats. So this tandem repeats occur in DNA when a pattern of one or more nucleotides is repeated and the repetitions are adjacent to each other. In tandem repeats, you find one or more nucleotides are repeated in the DNA which are adjacent to each other. These are tandem repeats. The arrangement of the histone genes within the cluster varies from species to species. Again, the histone genes arrangement depends on species to species. Short tandem repeats acts as caps for the DNA molecule and are crucial for maintaining the intact chromosomes. So they act as caps. The short tandem repeats act as cap for the DNA molecule and helps in maintenance of the chromosomal intactness. So these are tandem repeats. So within a DNA where you find the repeated nucleotides and these repetitions are adjacent to each other, it is called as tandem repeats. These tandem repeats act as cap to the DNA molecule and helps in maintenance of the intact chromosomes. Tandem repeats. Mini satellites and microsatellites. Mini satellites. These are located in the non-coding regions. And generally the size of mini satellites is 1 kb to 20 kb and repeat units ranges from 9 base pairs to 80 base pairs. In mini satellites, these are located in the non-coding regions and the size varies from 1 kb to 20 kb and repeat units ranges from 9 base pairs to 80 base pairs. Second type of tandem repeats is microsatellites. So these microsatellites are known as simple sequence repeats or short tandem repeats. These are generally 1 to 6 base pairs and microsatellites are used for the DNA fingerprint. Next one is interspersed DNA. First is tandem repeats and second one is interspersed DNA. A type of repeated sequence. Here you again see a repeated sequence, but the repeated copies are not adjacent to each other. As in tandem repeats, here the repeated sequences are not adjacent to each other. Again, there are two types of interspersed DNA sequences. The first one is transposable elements and second interspersed DNA sequences type are retrotransposons. So what are transposable elements? Transposable elements, it is a DNA sequence that can change its position within a genome, like it can jump within a genome. At any position it can change and sometimes it might create or reverse the mutations and it might alter the cell's genetic identity and genome size. So the transposable elements are generally the jumping sequences, the DNA sequences which can change its position within a genome. And while changing this position, this might create or reverse the mutations, alters the cell genetic identity and genome size. Transposition often results in the duplication of same genetic material. What are retrotransposons? Retrotransposons are a type of genetic component that can copy and paste themselves into different genomic locations. The transposable elements jump from one place to other, whereas retrotransposons copy and paste themselves in the different genomic locations. And they convert RNA back into DNA through the process called reverse transcription. So they generally convert RNA into DNA by a process called reverse transcription. So these are all other cells major and long entangled structures called chromosomes are formed, and after DNA duplication, you can see two sister chromatids are formed and which are connected to each other by centromere. What is a centromere? The connection in between is called a centromere, with which the two sister chromatids are paired, and telomeres are the structures which are towards the end of the chromosome, and these telomeres have two components like the repeated motif DNA and some special binding proteins. So there are two kinds of special binding proteins and other proteins. So these other proteins, they help in binding the chromosomal end to the territories of the nucleus. More compact organization of this metaphase chromosome take up the characteristic binding patterns after staining. So GSMA stains are used for this chromosomal binding patterns, where you can see the karyotype, and there you can see different chromosomes have different binding patterns. In this binding patterns, you find some darker positions in of G stains. So this represents AT regions. So this where AT is concentrated more, base pairs are concentrated more, there you see more GSMA stain. Genes are not only evenly spread along the chromosomes, but the density varies on the length of the chromosome. The density of the genes might vary among the length. The genes are organized into multi-gene families with similar or identical sequences. And eukaryotic genome also has certain genes called pseudogenes and evolutionary relics. So in among multi-gene families, you see ribosomal RNAs, you see pseudogenes, evolutionary relics, mammalian globulin proteins, mammalian globulin genes. So all these come under other genetic aspects. And the repetitive DNA content, it is again divided into transpositional and interspersed DNA. So the interspersed DNA. So this interspersed DNA has transpositional activity and tandemly repeated DNA. This includes mini satellites and microsatellites. So microsatellites are and mini satellites are involved in this, whereas mini satellites such as telomeric DNA and microsatellites generally used in the forensic sciences. Forensic scientists use this microsatellites in genetic profiling. So this is all about the summary. And for further references, you can read the books. These two books can be referred. And thank you for listening. And if you have any queries, you can contact me. Have a nice day.